topic of structure of chromosome. Chromosomes are highly condensed chromatin fiber. Chromosomes are readily picked up, or they have a capacity to pick up a certain dye. As they pick up a certain dye, the word chromosome has come into existence. The meaning of the word chroma is colored, while soma is body. That is why chromosomes are also referred to as colored body. When we talk about chromosome, the entire credit goes to Walter Fleming, who discovered chromosomes for the first time in the year 1882. He did this discovery in the larva of salamander. He observed that there were minute threads. These minute threads appeared during division, and these minute threads divided lengthwise. That is why he called their division as mitosis. Meaning is thread. Mitosis, thread. That was the original term that was used, or the meaning that was used. As on today, when we talk about mitosis, we say that mitosis is soma. That is the body cells. That's what we have already said. But here, the meaning that was given by Fleming is threads. When we talk about structure of chromosome, let's see what the details are. On the monitor, here is a diagram of chromosome for you. Chromosome is condensed and it is visible only during the process of cell division. When we talk about chromosome, it consists of two chromatids which are joined at a point called as centromere. You can see that these constrictions which are there are small constrictions. So, at this point, we should know that chromosome is condensed. Chromosome is the one that is visible only during cell division, very important. And chromosome consists of two chromatids joined at a point which is called as the centromere. It serves as attachment. When we are talking in terms of centromere, where is it attached? Previously, we have already studied that the chromosomes were attached to the spindle fiber and the attachment was with the help of the centromere. Each chromosome centromere is located at a particular site. As the spindle fiber contracts, the sister chromatids, they separate and move towards the opposite poles. So this whole process that takes place, we have already seen in cell division. As you can see, after completion of cell division, the chromatids, now called as chromosomes, they undergo a process that is called as decondensation, resulting in thread-like structures, which are chromatin fibers. These fibers will now reappear during next cell division. When we talk about the word chromatin, let us see the details of chromatin. The chromatin material that constitutes the fiber is formed of two substances, DNA, deoxyribose nucleic acid, which accounts to 40%. Second one, histone, a particular type of protein which accounts for 60%. The histones and DNA forms a kind of complex in which the DNA strands are wounded around a core of histones which are 8 in number. This complex structure that is formed is called as nucleosome. The entire chromatin fiber is coiled and supercoiled. To give you a comparison, this coiling and supercoiling looks more like your telephone cord. So, when you stretch it, it will be making that entire chromatin fiber straight. So remember the two main words. One is histones, second one is DNA. Structure that is formed is the nucleosome. At this stage, we have talked more in details about chromosome, we have talked about chromatin. We'll move on to the next segment. DNA structure and the shape was studied in details. Shape of DNA was studied by Rosalind Franklin in the year 1953. In the same year, Watson and Crick gave the structure of DNA. DNA is a large single molecule, hence it is also described as a macromolecule. It is composed of two complementary strands which wound around each other to form a double helix. Each single strand of DNA is made up of repeating nucleotides, which are made up of phosphates and sugar. The sugar is a pentose sugar. Along lengthwise arrangement that is seen, the nitrogen bases are attached to the pentose sugar on the inward side. The two strands are connected to each other with the help of hydrogen bonds. The complementary nitrogen bases are formed on the other strand. Thus, the two strands together form a ladder-like arrangement. The rungs of the ladder are the nitrogen bases. These nitrogen bases are guanine, represented as G, thymine, represented as T, cytosine, represented as C, and the last one, which is adenine, represented as A. It is always the complementary condition that is seen. Guanine is complementary to cytosine and thymine is complementary to adenine. We take into account the formation of new DNA. During interphase of cell cycle, each DNA molecule, that is chromosomes, duplicate for their equitable distribution in the two daughter cells. During mitosis, for replication, the DNA double helix, as you can see, opens at one end, making the two strands free, which can be seen. And the process continues with these strands becoming free and the formation of a new strand on the free strands that are present. This process continues lengthwise in the sequence of DNA, as a result of which there is formation of two daughter DNAs.